Hello everyone, welcome to this video lecture on viral zoonoses. So the last 10 years there are 5 major viral zoonotic diseases which are happening throughout this world. The first one is going to be avian influenza A H7N9 virus which happened in 2013. And the next year we had this Ebola virus, then we had this Zika virus, then in 2018 we have Nipah Virus. Finally currently we are facing this COVID-19. So in this lecture we can go through one by one. First one is going to be avian influenza A H7N9 virus. So as the name implies, this is going to be caused due to your birds. So it's going to be a subtype of your influenza virus. So in human, it's first reported in 2013 in China. So after this first thing, there's going to be a 5 waves of epidemics which happened due to this virus, out of which more than 1,500 human cases are infected due to this virus, out of which 40% resulted in the death. Luckily, due to this computational as well as these genetic studies, they identified this sequences which are sensitive to this particular antivirus. So which is going to be a neuroamynase inhibitors. So this inhibitors it's going to be preventing your viral thing. So these antivirals it's going to be osteomide and the second one is going to be zenamide. So though these two things are used for the treatment, the effectiveness of your osteomide is still under question. Why is that? Because the patients who are administered with this osteomide have seen that they're building up the resistance against this particular virus due to the usage of the drug. That's why this thing is still under question. We need to understand how this particular virus is being evolved. So for that we need to understand this multiple reassortment events. So first let's understand what real assortment is. So it's going to be mixing of the genetic material of the particular species. Thereby we are going to have new combinations. So out of these new combinations it's going to be getting onto this particular individuals. So here we are going to be seeing an example with the H7N9. So we have first this domestic duck where the genome is going to be represented in the blue. Next we have this wild birds where it's going to be mentioned in the green, and the last one is going to be a domestic poultry which is mentioned in pink. So in case of your H7N9 virus, we are going to have this combinations of all these particular individuals. So first we have this HA which is going to be the hemoglobin which is from this domestic duck. And then we also have this green which is Na due to the wild birds, and then because of this domestic poultry we are going to have this more kind of genome from this particular individual. It's going to be a combination of all these genomes where the most of the characteristic features comes from the domestic poultry. So then this particular virus is getting to this individual. So the next event which I'm going to try to understand is going to be a genetic tuning. So as we can see before, here also it's happening that continuous amino acid substitutions which is happening, because of which there's going to be adaptations which are happening. So in the previous cases as we saw before, it started in this wild birds as well as domestic things and then getting to the virus. So how it's going to affect in the future is that this genetic tuning might cause further adaptations in this particular weight of virus which might be much more pathogenic when compared to the existing one, and then the pandemic might be it's going to be severe than the existing one. We need to be prepared that how this genetic tuning is being happening so that we have an idea upon which we can develop this antivirals accordingly. Next one is going to be Ebola virus. The outbreak it happened in the West Africa in 2018. And out of it existed for 2.5 years. There are going to be 2000 28,600 cases, and then out of which 11,325 deaths have been happening. Luckily, we had this vaccine for this particular Ebola viruses, which is being developed by Merck, which is a US-based pharmaceutical company. So they developed this vaccine. The drawback of this particular vaccine is going to be it's going to be specific to the particular strain of this Ebola virus. It's not for this all the strain. We can see much in detail further. So let's try to understand how the drug which is developed against this Ebola infection it happens. So any virus when it get into this human cell first it need to interact with this cell membrane. So if we prevent these interactions, we can overcome this virus infection. So here the first step is either if we use this antibodies or the particular chemical inhibitor, we can prevent this interaction of the virus. That is not going to get in the cell. Even it happens, then if we use another component such as EIPA, it's going to prevent your macroprimosis inhibition, whereby your virus is not going to get into the vesicular membrane, thereby the entry of the virus it can be prevented. Similarly, for each event, if we use a specific component, then we can prevent this virus entry into this cell, this is a host, thereby we can prevent the replication. Next, we're going to see this Ebola vaccine. So, the Ebola vaccine that is developed, that is approved by FDA in 2019. So, the trade name of this Ebola virus is going to be Ervebo. As we mentioned before, it's going to be marketed and developed by this Merck. So we mentioned that it's going to be active against a particular strain which is going to be the Ebola virus. So why? It's because it's that this vaccine is developed based on the strategy of a live attenuation. That means this particular virus is genetically modified that it's not that much pathogenic, but still when it is administered into the particular human, immune response is developed against the particular pathogen. That's why it's going to be specific against only this particular strain. In order to overcome this drawback, 7 other Ebola vaccines have been developed with different strategies. Mainly, it's going to be your nucleic acid based vaccines. Why? Because is that if we have this nucleic acid based vaccines, such as your DNA vaccines or RNA vaccines, it's going to be much more broader. That means it's going to be effective against different strains of your Ebola vaccines. Next one is going to be a Zika virus. So the epidemic is started in 2015 in North and South America. In 2016, the nation reported 5,168 cases, which mostly due to this patients they traveled across the US. So this Zika infection happens due to this bite of the particular species of the mosquito, which is going to be Aedes species. Unfortunately, we don't have a vaccine or medicine against the particular Zika, so we need to discuss about this in detail. But first, let's try to understand the life cycle of the Zika virus. So as we can see here, we have this host cell, and then the Zika virus it needs to get into the host cell. First, it's going to be trapped, and then it's going to be getting into this particular vesicle. So this vesicle is kind of happens due to this endocytosis. In endocytosis, we have different types based upon the protein interactions or the lipids which are present upon the vesicles. So here it's going to use Catherine median endocytosis. So if we use a particular chemical component which is against this one, then we can possibly develop an antiviral against it. So similarly, when this particular virus is getting into the cell, it's going to be cleared out of this endosome, and then it's going to be released into this cytosol of this cell. So when it's released in the cytosol, this nucleic acid components it's going to undergo this translation, whereby the protein components of the virus it's going to be formed and then it's going to be assembled, whereby one virus it's going to become many viruses and then it's going to be released out of the cell. This slide it shows the Zika virus infection and potential side effects. So when this Aedes mosquito, when it bites the particular patients, this virus is going to get into the skin. In the skin, we are going to have this keratinocytes. In addition to keratinocytes, we are going to have this immune cells. Mostly your antigen presenting cells such as your dendritic cells which is going to take up this virus, and then specific immune response is going to be generated against such particular virus. Though there's kind of immune inventions happening, few viruses, it's going to this bloodstream. So this point where this virus gets into his bloodstream, that we call it as viremia. So this, in this case of this viremia, it's possible that three things can happen. So again, if a mosquito bites this infected person, then this is going to be containing this transmission of this particular virus from this patient to a new mosquito. That is going to be first possibility. The second possibility is that in case if the patient, if she is a female and if she carries a baby, then there's possible that there a possibility of transplacental transmission, and then the child also whether it has, that needs to be tested. And then if there is a sexual transmission, that also needs to be tested. And then when it is present in the blood, obviously it needs to be clinical characteristics. It might lead to fever, rashes, conjunctivitis, etc. So whether these components it's going to convert into adverse effects, that also might be seen, and what are the components that are responsible, that also needs to be studied. And finally, we also need to see that whether this thing it affects the neuronal system. Thereby we can develop this antivirals. Also these other components can be developed, thereby we can prevent this severe side effects of this Zika virus infection. Next comes your Nipah virus. So this outbreak is first happened in 1999 in Malaysia. So it happened in the fifth workforce. Then after several years in India also it happened in 2018. So out of this 2000 people are quarantined, and then they struggled to overcome this outbreak. So this happened due to this few bats of the Petropodia family. They are going to be a natural host of the Nipah viruses. So as you mentioned, this way of the bat is going to be a natural host, and the Nipah virus, when it is present in this bat, it's going to be asymptomatic. So when this bat, if it consumes a particular fruit, and then when this fruit, if it is consumed by this particular pig, then the virus is going to be transmitted from the bat to the pig. In addition to that, it's also possible that the waste also can be consumed by the particular pig. However, the pig, it has the virus. So when this pig has a virus, it might be experienced with mild to severe illnesses, and it's possible that transmission between the pigs can also happen. When the secretions of this pig, if it comes in contact with the humans, or there are people who consume the meat of this pig. If they consume the meat of the particular pig, but that also they can get this particular virus, thereby they can have this illness. It's also possible that human to human transmission can also happen in this Nipah virus. Researchers have studied the structure of this Nipah virus, through which they found out the inhibitors through computational studies. So they identified 4 putative peptide inhibitors and 146 small molecule inhibitors against the Nipah virus proteins. So this Nipah virus proteins, as we can see in the diagram, so there are proteins such as a G protein, which involves in the attachment of this particular virus into the host. Similarly, we have this M protein dimer, which helps for your budding. Similarly, we have these other proteins which helps for the capsid assembly, genome replication, and fusion. So if you have an inhibitor which affects this particular characteristics of this protein, then we can prevent the replication of this particular virus in the host. This is the overall ideology which is happening. So out of this 150 inhibitors, they have shortlisted 13 based upon this binding strength, stability, and effectiveness against multiple strains of this Nipah virus. Thereby, it can be broadly effective. Then comes your coronaviruses. Currently, we are facing with COVID-19. Normally coronaviruses, they are going to be a non-segmented positive sense RNA viruses which belongs to the family of coronaviridae. So there are several types, alpha, beta, etc. Out of which the one which causes the epidemic is going to be the two kinds of beta viruses. First one is going to be here SARS-CoV, which is going to be severe acute respiratory syndrome coronavirus 1. Now we have this internal case of SARS-CoV-2. And also they had this Middle East respiratory syndrome was coronavirus. So the death rate of this one is going to be 10% for your SARS-CoV and 37% of your MERS-CoV. In case of your SARS-CoV-2, the death rate keep on it's going to be increasing. So why is the case? Because of this SARS-CoV-2 is adapting and mutating continuously, thereby it's difficult to control the spread, and then the treatment is going to be difficult. So in our channel we have other videos which particularly focus upon your immunomodulation of these viruses. They will be also be discussed about how the virus entries. We also have this genome studies and etc. If you are interested, please go through those videos for the detailed information. Thank you. If you have more questions, please email to this ID which is mentioned here.